let's get into how does this shorten? How does a muscle contract? Okay. So first we need an action potential. This is a signal from a nerve or a neuron, okay, that tells the muscle to contract. Okay, so again, this is known as an action potential. So this is excitation-contraction coupling. So excitation-contraction coupling are the events that lead up to a contraction and then the contraction itself occurring. So the whole process, okay? So the action potential is sent along the sarcolemma here, okay, down into the T-tubule area, and that's where it stimulates the sarcoplasmic reticulum. What I didn't go over is in the sarcoplasmic reticulum, we have calcium stored, okay? The release of calcium is what leads to a contraction, okay? So how does that happen? Remember we talked about these two proteins here, troponin and tropomyosin? Well, how calcium helps a contraction to occur, calcium binds to troponin, these green areas here. They cause a conformational change, which is a shape change, and they move the tropomyosin out of the way. Remember I said we had these myosin binding sites on our actin here? So again, calcium binds to troponin. Troponin causes a conformational change that moves tropomyosin out of the way of those myosin binding sites. And now the myosin heads can bind to actin, or thin filament, and a power stroke occurs. So it pulls on the actin, okay? And then it can bind again, pull on actin, bind again, pull on actin, okay? All right. So the action potential is very quick and it's gone before the actual contraction occurs. Okay, so let's talk about exactly how this action potential thing happens. Okay, so we have the neuromuscular junction is the area where the nerve interacts with or where the neuron interacts with the muscle. Okay, so let's say our neuromuscular junction, we have our synaptic end bulb here, okay, or let's draw it like this. Okay, so we have our synaptic end bulb here interacting with our muscle fiber, okay? So what happens first is in the synaptic end bulb of the neuron, okay, there are these channels. They're voltage-gated calcium channels. So it's these little channels here. Let's draw a couple, okay? So they're closed until there's a certain voltage, which would be caused by a signal sent down this neuron. Once that signal tells these to open, they can open up, okay? Okay. When these open up, it causes an influx of calcium. So calcium enters into the neuron here, the end bulb here, okay? And this causes neurotransmitter to be released. So now we have neurotransmitter, in this case for action potential, it's acetylcholine. So now acetylcholine is in the synaptic cleft, which is the space between, in this case, the neuron and the muscle fiber, okay? Now, here on our motor end plate, or our sarcolemma here, we have something else going on here, okay? We have sodium channels, so ligand-gated sodium channels. So I'll draw in green some sodium channels here, and let's say they're shut. Once this calcium enters here, or once the neurotransmitter enters into this synaptic cleft here, the sodium channels open up, okay? And acetylcholine can, acetylcholine will cause these to open up, and then sodium will enter, that's in this space here as well, will enter into the cell, okay, into the muscle cell. So sodium will enter in here. Okay. This depolarizes the muscle, so it causes the inside to become more positive as we move down the sarcolemma here, and etc. So as we go down, it becomes more positive. Okay. and it results in calcium being released from the sarcoplasmic reticulum. So let's draw our calcium molecules in here. Okay, calcium in here, our other sarcoplasmic reticulum, and of course we have many of these as you keep going down the sarcolemma. Okay, so there's actually, you don't need to know the names of them, but how this happens is there's two proteins here. I just want to explain it a little bit, even though you don't need to know in this much quite detail. There's these two proteins. There's a protein here and a protein here, okay, that are kind of bound together. As this depolarizes down the T-tubule, this protein pulls on this one and it causes this gap here, and now calcium can rush out of the cell. Okay, so that's how that happens there. And then again, calcium can bind to troponin, make it have a conformational change, move tropomyosin out of the way. That frees up the myosin binding site, and now the myosin heads can bind to those myosin binding sites, and that power stroke can occur, or pulling in, or muscle contraction. Okay. Okay, so there are different ways that the muscle gets the energy it needs, okay? So what happens when the myosin binds, okay, onto the thin filament or onto the actin is through ATP, okay? How it releases is really through ATP, okay? So our contraction cycle is actually in a little bit more detail, right? The myosin heads here, they hydrolyze ATP, so it becomes ADP. So there's an ADP attached here. So one phosphate comes off and we go from ATP to ADP. Okay, so ATP, right, we went over this a little bit in unit 1. ATP is adenosine triphosphate. So there's 3 phosphate groups. I'll just draw them out like this. ADP is adenosine diphosphate. So one of these comes off here and that releases energy. Okay, and we have ADP. So when ADP, or when ATP is hydrolyzed, that's when our myosin heads become energized and they get ready in position, okay, to pull on those actin heads, okay? Then they bind to actin, power stroke occurs, the pulling in occurs, okay? And once it's bound, how does it become unbound? ATP comes back in and binds, okay? And that actually releases the myosin heads, okay? So ATP is actually what releases the myosin heads. But we need energy for this to occur in either case, okay? So how do we get energy? So you don't need to know the full processes of these things, but you do need to know the 3 ways that we can derive energy for muscle contraction and how long they provide energy for, or how much ATP they provide, okay? So the first is creatine phosphate, okay? So in creatine phosphate, creatine kinase, which is an enzyme, catalyzes the reaction to transfer a phosphate from creatine phosphate to ADP to quickly get ATP, okay? So it only provides about 15 seconds of energy, creatine phosphate. So that doesn't provide us too much energy, but short bursts of energy. Then we have anaerobic glycolysis. So anaerobic means no oxygen is needed, okay? So when we're done with our creatine phosphate storage, glucose can be converted into pyruvic acid and it generates ATP. This gives us about 2 minutes worth of energy. Okay, so it gives us a bit more, but still not that much energy, right? And then there's cellular respiration. So you probably learned about this in your biology courses, but this is the Krebs cycle, okay? So this uses oxygen, so it's under aerobic conditions. Pyruvic acid can enter into our mitochondria, which give us ATP. and undergoes a bunch of reactions that require oxygen to generate a large amount of ATP. Okay, so 30 to 32 ATP molecules, whereas the other two you only got about 2 ATP molecules. Okay, so this can give us up to hours of energy. Okay, cellular respiration. Okay, so that gives us the vast majority of our energy. Okay. All right, so our muscles, of course, can get fatigued. Again, for our cellular respiration, we need oxygen. Okay, so we can get tired because of depletion of oxygen or depletion of creatine phosphate or other nutrients that we need for those processes. Our muscles can get fatigued if there's not enough calcium being released from the sarcoplasmic reticulum. That would cause if there's not as much calcium, it cannot bind to troponin and move tropomyosin out of the way for the myosin heads to bind, and then we can't have as many contractions. Okay. Buildup of ADP, adenosine diphosphate, or insufficient release of acetylcholine at the neuromuscular junction, then we don't get this depolarization to cause the contraction in the first place. Okay, so those are ways in which our muscles can get fatigued. Okay, so a muscle contraction has different strengths, right? We can kind of use more like finesse, like picking up this pen here. I don't need to use a ton of muscles to pick up this marker, right? Or I could do powerlifting, right, and lift something very, very heavy. Then I need to use a lot more muscles and different types of muscle fibers, okay? Okay. So let's talk about how our motor units or our muscle fibers are recruited, okay? So our weakest motor units would be recruited first, and then we would recruit stronger and stronger based on the strength we need for something to occur, okay? And then our muscle fibers kind of take turns contracting so that we can have a more sustained action, okay? So if we're at the gym for an hour, then we're using different muscles and they're kind of taking turns and replenishing themselves so that we can keep that stamina up. Okay, let's talk about the muscle twitch. So there's 3 phases of a muscle twitch. We have a latent period. This is where we have our excitation-contraction coupling. There's no muscle tension seen yet, so this would be like the action potential occurring. Then the period of contraction, this is where the cross-bridge formation happens, so where myosin binds to actin. Okay, so tension increases. Okay, and then we have our power stroke or our contraction. Then we have our period of relaxation. So this is where calcium enters back into the sarcoplasmic reticulum and our tension declines back to 0. Okay, so the muscle actually contracts faster than it relaxes back. Okay, so we have different frequencies of stimulation of the muscle as well, okay? So we have what's known as wave summation. This happens when a second action potential triggers the muscle to contract before the first contraction has finished. And they'll kind of summate or add up, right? A sum is an addition of something into a stronger contraction, okay? So with wave summation, we have 2 types, unfused and fused tetanus, okay? So unfused is like an incomplete tetanus. There's a smooth, continuous contraction, and those add up. Those are the ones that summate. If the stimuli frequency continues to increase, muscle tension can reach a maximum known as fused tetanus. So those contractions kind of fuse into one smooth and sustained contraction. This is very rare, okay, fused tetanus. This is like when you hear those stories of a mother like lifting a car off of their child. That would be like when a fused tetanus would occur. But it doesn't happen in everyday life by any means. Okay, so let's erase all this and we'll draw out those. Okay. All right, so over over here we have our force of contraction. So I'll just write force. So that'll be our force of contraction. And then we have our different scenarios here. So this here would be our single twitch. This would be maybe like maintaining our posture, like those small things that happen. We would have one action potential and we'd have a little force of contraction, okay? Wave summation, okay, would be we have a signal and then another signal and it adds up to a little bit of a stronger contraction there, okay? Unfused tetanus, we have let's say 4 signals in this case. So 1, 2, 3, 4, and it adds up to this stronger contraction, but we're still not anywhere near our maximum there, right? Our fused tetanus, we would reach the maximum. So we would have many action potentials. So one right after another, after another, after another. So very, very quick. And they just raise so quickly to our maximum and we plateau here. And then at some point we drop off when we're done with that activity, okay? So this here would be our fused tetanus, unfused tetanus, wave summation, and here's our single twitch, okay? Okay, so we have different types of contraction. Isotonic contraction, we have a constant tension and the muscle length changes. In an isometric contraction, the muscle contracts but does not change length, okay? So our 2 isotonic, we have concentric. So concentric contraction would be like picking up a book. So the muscle is going to shorten as we lift a load, okay, or something with weight, okay? Eccentric contraction is like lowering a book. So our muscle is actually lengthening, but we're still doing work, right? It still takes some power to lower something down if you're holding something with weight, okay? And then our isometric contraction would be like holding that book out in place, okay? Isometric, so holding that book out in place. So you'd obviously get tired. So in this case, I don't have it drawn out anymore, but our myosin heads would be bound to actin and they would just be kind of like spinning their wheels. They're like ready, but they're not gonna cause a contraction. So you're just holding something out and they're ready, they're doing work. But you're not actually shortening or lengthening the muscle in that case, in an isometric contraction, okay? Okay, so we have different muscle fiber types as well. Let's briefly talk about those. So we have slow oxidative fibers, slow oxidative, okay? In this case, this would be for low endurance or low intensity endurance activities, okay? So this would be like for maintaining our posture, right? We can kind of do that all day long without getting too tired, okay? So these are things that are longer term. Okay, but low intensity, not like running a marathon, right? Fast oxidative fibers. Okay, these are for medium intensity activities. So this would be like walking or maybe sprinting somewhere a short length. Okay, and then we have our fast glycolytic fibers. Fast glycolytic. These are for short-term bursts, so short-term very intense or powerful movements. So like hitting a baseball, right? So something that's very short-term, but you need a lot of force for, okay? Have you seen people do like the shot put? It'd be like shot putting as well, okay? Anything where you need a lot of force for just a very short period of time, you would recruit these fast glycolytic fibers, okay? So you would recruit, remember I talked about motor recruitment? You would recruit these slow oxidative fibers first, then your fast oxidative second, and then your fast glycolytic third, okay? And different people may have different percentages of these different types of fibers, types of muscle fibers, depending on what you do. So if you've been a baseball player your whole life, you'd have more fast glycolytic fibers than someone who's a sprinter, who would have more fast oxidative fibers, versus someone who's more maybe sedentary would have many more slow oxidative fibers. Okay, and then let's briefly talk about cardiac and smooth muscle. So cardiac cells, we talked about cardiac muscle a little bit, so you could read through it in the PowerPoint, but some things I didn't mention when we went through histology. Cardiac muscles have more mitochondria, and their contractions last a lot longer than skeletal muscle contractions. So cardiac muscle is very important because it helps to pump blood throughout our body, which gives nutrients and oxygen to our tissues. So it needs a lot of energy to maintain that. Okay, and then smooth muscle, this is surrounding our other organs. Okay, and it looks a lot different, right? It has those flattened cells, or smooth, there's no striations there. Okay, and smooth muscle contractions start slowly, but they last longer than both skeletal or cardiac muscle contractions. Okay. Okay, aging in muscle tissue. So between 30 to 50 years old, about 10% of our muscle tissue is replaced by fibrous connective tissue. Okay, so of course, if our muscle tissue is replaced by connective tissue, we're not going to be as strong, be able to do the same things we used to do when we were a bit younger. Okay, and between 50 to 80 years, another 40% of muscle tissue is replaced. Okay, so we have, again, lower muscle strength, slower reflexes, and our slow oxidative fiber numbers start to increase as we age.